human beings are all alike. They are also all different. Most differ just slightly. Color of hair, eyes, or skin, for instance. But a few differ drastically from the normal, and it's important to know why. The inherited differences in human beings are difficult to study because it takes too long to gather data on just a few generations. Geneticists have found one way to get around some of the problems. That's to study simpler forms of life that multiply rapidly, like bacteria. Dr. Norton Zinder is a geneticist and a member of the faculty of the Rockefeller University. He was in charge of the genetics experiment covered in this report. The bacteria in his lab are called E. coli bacteria. They're so small that even at a magnification of 1,000 times, little detail can be seen. They split in two, giving birth to a new generation every half hour. And in one day, scores of generations live and die. E. coli are common, harmless organisms found in water supplies, in sewerage systems, and in the intestines of human beings. This is the most they can be magnified with the light microscope. But photographs taken with the electron microscope at magnifications of close to 100,000 times reveal many more details. Among the things that can sometimes be observed inside the bacteria are small particles of matter called viruses. These particular viruses, called F2 viruses, are the smallest known self-replicating organisms on Earth. They were discovered by Dr. Zinder. The F2 virus is a parasite that grows inside bacteria. As a parasite, it gets along in life with very little to do except to make 3 known proteins. More complicated living things make millions of different proteins. The F2 gets by with just 3. One of these, the coat protein, surrounds and gives shape to the vital genetic material that bears the inherited instructions for its life, called RNA. The life story of the virus is quite simple. They first attach themselves to the hair-like structures on the outside surface of the bacteria. The viruses shoot their RNA into the bacteria. Once inside, the RNA then directs the making of coat protein and complete viruses are formed. RNA has the ability to reproduce itself. The viruses multiply rapidly, one virus producing close to 20,000 offspring in an hour. There are more F2 viruses in the world than all other organisms combined. They use up the resources of the bacteria until finally the bacteria may be literally consumed by millions of viruses. They then go out to search for other bacteria until there are no more bacteria to devour. Geneticists use the F2 viruses because they're harmless to higher organisms, such as geneticists, and because their life functions are so simple that they can be isolated and studied in detail. In addition, the viruses themselves can be torn apart and things done to them that can't be done to more complicated forms. Our social conscience places no limits on what a geneticist can do to a virus. But to get back to the experiment, Dr. Zinder became interested in a report from another lab that a mutant virus, while it grew in most strains of bacteria, did not grow in one particular strain, called the non-permissive strain. The mutant virus would evidently send its genetic material into the non-permissive bacteria, but no new viruses were formed. It was reported that the mutant, instead of making its full coat protein, made only a small part of it. The assumption was that a genetic word in the middle of the mutant's message for coat protein was being read as stop, do not make more protein, instead of go, complete the protein. Dr. Zinder requested and then received a sample of the non-permissive bacteria from the geneticists who'd grown them, along with a normal control type. He hoped, using a mutant of his F2 virus, to experimentally confirm and extend their work, and to determine how the word for go in one organism meant stop in another. The experiment that followed dealt with events beyond the range of vision, beyond the range of any microscope, the making or not making of coat protein. Can we understand these events that can't be seen? Well, perhaps we can understand some of them by listening to a discussion that could have taken place before the experiment began.
Now, the F- strain phosphatase appears. It doesn't. It has a mutant phenotype in the male strain, in the donor strain, and it becomes phosphatase positive in the female strain.
Well, I'm afraid that didn't help us very much. It wasn't possible to understand most of the language of the geneticists, but it was possible to establish the fact that the experiment needed planning, that the results were not at all assured, that it was to involve a number of scientists with widely divergent views, and that it would take a long time.
Well, if we can easily test 500 of those in a day, no problem.
We only need one or two.
The ones that you want to look at. Once we get the mutant, I think we'll probably screen, 3,000 before I'd be willing to quit. After a good dose of nitrous acid, I would say that at a minimum we pick 3,000 to 4,000, and necessarily everyone will take a turn.
All right. I can't say anything else but to try it.
The first step in the experiment was to take the F2 viruses in the lab and to alter them to make future generations of mutants that would grow in one strain of bacteria but not in the other. The way to alter genes is to use either X-rays or chemicals called mutagens. The effect of the mutagens, in this case nitrous acid, is to kill off most of the viruses. A few of the survivors are mutants, and then these few have to be sorted out to find the particular mutants that will grow coat protein in one strain of bacteria, but not another. After the F2 viruses were treated by the nitrous acid, they were placed along with bacterial hosts in a supporting medium, which had food for the bacteria. When this mixture is put on a plate, the supporting medium hardens to form a semi-solid base. The bacteria are grown in an incubator where a constant temperature, close to the temperature of the human body, is maintained. Since this growth takes 6 hours, we've taken time-lapse films to compress the hours into 1 minute. We can see the bacteria on the plate as they rapidly grow to large colonies. The viruses then begin to grow in some of the bacteria. The viruses cannot be seen, but the effects of their growth are visible. As they devour the bacteria, they clear out an area of the plate, leaving behind empty spaces that appear on the plates as dark holes.
Nieti, excuse me. Would you please check these? We're looking for mutants that will grow on the permissive strain and not on the non-permissive strain.
Yes.
In the 6 hours, a dozen or so generations of bacteria and viruses have been born and have died. The search for a mutant begins. The areas taken over by the viruses can be easily seen as black spots and picked off the plates. At this point, no one can tell if there's a mutant among them or not. In all of genetic history, it has always turned out that the study of mutants has led to the most important discoveries. There are rules to the genetics game that we don't know. The mutant says, in effect, I'm outside the rules. I've got one change, one difference, and that's enough to make the system look at me and reject me. Why? If we discover the why, the mutant's difference then leads back to where we want to go, to an understanding of the normal. An assembly line system was set up whereby a number of the viruses could be put on plates at once. Each group of viruses was plated. First in the permissive bacteria. The same group was then plated in the non-permissive bacteria. The offspring of a particular virus was therefore to be found in one spot on one plate of permissive bacteria and on the same corresponding spot on another twin plate of non-permissive bacteria. The plates are again put back in the incubator where generations of viruses and bacteria were to reproduce, except for possibly one virus on one of the plates that would not. Six hours can be a very long time or a very short one. It's a long time when you're waiting for results, but a short time to get a dozen or so generations of living things. After the 6 hours, it was necessary to visually screen the plates for an area of bacterial growth where the virus had not consumed the bacteria. There were hundreds of plates to be examined. Normally, one type of mutant might occur spontaneously in hundreds of thousands of viruses, spanning many generations. Because of the nitrous acid treatment, there might now be one mutant in 1,000 in a few generations. The question was whether or not this one mutant could be found. One day, an area where the bacteria were not destroyed, where there was no evidence of virus growth, was discovered. This result had to be checked against the twin plate to confirm the growth of the same virus on the other bacteria. There was an area of virus destruction on the twin plate, and in this area, the one mutant virus and its offspring were finally discovered.
Dr. Zinder?
Good morning, Dr. Zinder.
Did we get any mutants?
I have one here, which is out of 500.
Let me see it, please. Oh, that one is extremely promising. Extremely promising. Do we have any bacteria grown up so that we can make a stock of this immediately?
Yes, we do.
Oh, very good, very good. Let's do that, please.
It was relatively easy to grow many new generations of the mutant in a flask full of bacteria. Finally, a whole strain of laboratory-bred mutant viruses that would not grow on one strain of bacteria, but would grow on the other, came into being. The new strain of mutants was tested periodically to make sure that they were growing as they should. Again, they were to be grown in the incubator and then screened for the telltale dark holes on the plate. Individual viruses cannot be counted, but the billions of their descendants can be, as a group. Geneticists describe what billions of viruses do or do not based on studies of whole populations rather than the study of individuals. Having successfully grown a strain of mutants, it was now necessary to complete the second half of the experiment, to go from events in the plate to events in the test tube, to go from what scientists call in vivo, meaning in life, to what they call in vitro, meaning in glass or in the test tube. This, again, as in all scientific matters, called for a discussion.
Because, you know, the in vitro system is a fairly inaccurate system. And so you've got some, I mean,
Well, how do we know?
In the main that it's inaccurate?
We don't know. We have a system that can even prove that it's accurate.
Well, you can
If
prove
it-
that
look,
you
we-
make code, but what if 10%
all right.
of the times you read a codon, you misread it? You wouldn't pick that up in a tryptic.
Okay, well, I mean, maybe that's it, or maybe what you're misreading, well, what you're saying, the other things you read, maybe actually are the other systems. If it is a polygenic message, I mean, all right, we're really getting speculative now. We don't know.
A lot of other people working on the fidelity of the
After all we
system.
know is somebody, you take E. coli and you split it open and you throw an RNA and you get something that looks like it makes.
Right, and I think we've looked at most of the peptides that are made, and they seem to have a fairly reasonable degree of fidelity. I would say that the in vitro system is surprisingly faithful.
It was decided after discussion to try to extract the pure RNA of the mutant and then actually test its ability to make coat protein in the test tube. A centrifuge was used to spin the concentrations of viruses and bacteria a number of times to take off the heavy bacterial walls, the cell debris, the other extraneous matter, until finally the viruses were all alone by themselves in the very thin middle layer of the test tube. The viruses were then taken apart by a process of chemical extraction. The coat protein of the virus is soluble in the chemical extract and therefore goes into one layer. The pure RNA of the virus remains in the water layer. This separation is not complete until it is helped along by being shaken up to bring everything in contact with everything else and then spun off again in another centrifuge. Finally, coat protein and RNA. The RNA is removed from the coat protein. Then the water is removed from the RNA. And what remains after all the separations and extractions is the pure RNA of billions of viruses. To see the RNA of just one virus, we need the help of the electron microscope. This electron micrograph was taken at a magnification close to 100,000 times. Normally, this RNA is coiled up inside the virus, but here it's unwound, and we see that it has a long thread-like structure. Within this thread, and unfortunately hidden from view, is a long chain of chemical compounds that bear the code for its life processes, making 3 proteins. These instructions are strung out, one piece of chemical information after another, in the long thread. This system of coding is the same for every living thing on Earth: virus, bacteria, fish, monkeys, and men. To put the RNA to work making coat protein, a mixture was first prepared of water, food, stabilizing agents, buffers, and all the amino acids but one.
Very good. Now all we want is 0.25 mils of the amino acid mixture minus phenylalanine. And then we'll be all set to start the reaction. Now on both of them, we add 0.225 mils of water. 0.225. 0.23 is fine.
After this was all brought together, the non-permissive bacterial extract was added.
Fine. All right, we'll start the sus3 first. Okay, Pete, Norton will be ready in about 30 seconds. Okay, wait a minute. I'll add the label first, Wilmer. At 30 seconds after, and then you the RNA.
The missing amino acid that must go into the production of coat protein had a radioactive tag added to it. The coat protein will therefore be identified and measured by the amount of its radioactivity. The last item was the RNA of the mutant in one tube, and then the RNA of the wild type in another. As soon as the RNA hit the tube, the production of coat protein began.
Next one, Wilmer. Okay, Pete, Norton. And you have to start taking points. Okay, ready.
After a few minutes, the mutant mixture was drawn off every 20 seconds, and then the wild type drawn off every 20 seconds.
See, Phi is one of the beginning amino acids, isn't it? I think so.
Mm-hmm.
Right. So if it's small, we'll pick it up with Phi. Lysine doesn't show up till late. At least this will show us the difference in the big peptides.
The result was one set of tubes with the coat protein production of the wild type, and another set of tubes with the coat protein of the mutant, if there was any.
Well, let's go. We're falling behind.
You?
Yes.
Here are the samples from the incorporation we just did.
Oh, good.
This is the mutant, and here's the wild type. Would you get these
Oh,
filtered
good.
as soon as possible?
Sure, yeah, I'm just as interested as you are.
Okay.
It'll be about 40 minutes.
Okay, then. Very
That'll
good.
be
Let us
kind.
know.
Certainly will.
The key to this part of the experiment is to take something that is tiny, like the amino acid with the radioactive tag, that will go through a filter, and to try to make it into something big, like coat protein, that will not go through the filter. It's so simple, yet the understanding and the interpretation of the results is so difficult. First, the tubes of the mutant are filtered, and then the wild type. The fiberglass filters are quickly dried with infrared bulbs, then placed in bottles. This is the easiest way of handling the filters. The amount of coat protein that has been made, and therefore that stays on the filter, can be counted in a radioactivity counting machine. The counter tells you if you're in business or not. It's the payoff. A high percentage of all the results are shown on this counter. The results in this case showed that the wild type had made a great deal of coat protein in the test tube. The mutant, on the other hand, had made very little by comparison. The samples were then put through another kind of filter called the Cephidex column. This column has beads inside that can sort out the proteins made in the test tube. Because these proteins are submicroscopically small, they have never been seen. We will illustrate their theoretical motions through the beads with animated drawings in which the proteins have been enlarged millions of times. As the proteins enter the column, they differ in size. When they reach the beads inside the column, the smaller proteins enter the beads and then move around inside through many internal passages. The larger proteins, such as the full coat protein, are too large to enter the beads and therefore have the shortest and fastest path to the bottom of the column. They reach the bottom before the small proteins, which turned out to be a small fragment of the coat protein made by the mutant virus. The samples were taken off the column at prescribed intervals. First the large samples, the full coat protein, then the small samples, the fragments of coat protein made by the mutant virus that were not big enough to permit the mutants to form complete viruses in the non-permissive bacteria. After more counting and computation, they knew the exact size of the fragment the mutant was making and how much it was making. From this, it was possible to tell where the stop sign had been misplaced in the genetic code of the virus. Finally, they were able to adjust the chemical composition of the extract from the non-permissive bacteria and test it with the mutant RNA.
Okay. To the sus3 tube.
And after filtering, more counting. The stop sign now read go. The mutant RNA now made full coat protein in the new extract from the non-permissive bacteria. The geneticists now knew more about the two strains of bacteria, the permissive and the non-permissive. They also knew more about the nature of stop signs in viruses, and possibly more about the stop signs in the genes of all organisms. The experiment was complete. To sum it up, we would say that it began with the work of many other scientists in other laboratories. It was carried forward step by step with the use of imagination, logic, and relatively simple devices that explained in a most profound way the secret workings of the genes that lie hidden in tiny viruses and in all living things. It was completed by the joint effort of a group of people rather than one.
It's still going up. It looks like it's approaching. We don't have the S on it.
How would a geneticist sum up the experiment? We asked Dr. Zinder.
When you work with microorganisms, as I do, there's a bit of a feeling of being far removed from reality. When you look at the world around you, you suddenly realize that the things you've learned about microorganisms are really occurring and going on right in front of your eyes. There is an essential unity to all of living matter. Based on the knowledge that we've gained with bacteria and viruses, it'll probably be possible to understand the genetics of man and perhaps even to benefit him. I don't know if it'll take 50 or 100 years, but it really must occur.